This is gonna be one of my favorite series to teach you guys. See, the great topic is jaundice. Under this topic, there are many subtopics that you need to know. But the problem is that these subtopics are present in different subjects. And the problem with university lectures is that these topics are taught in different subjects at different lectures at different times. So, integrating the concept from these topics becomes really difficult. What I'm gonna do is take all these important topics and then teach you them in a series so that the whole picture of jaundice becomes crystal clear. For example, you know this much that jaundice has something to do with bilirubin. So first you need to know what is bilirubin and where exactly in our body is bilirubin produced. Now that's a topic of biochemistry. Then you need to learn about hyperbilirubinemia, congenital and acquired, along with prehepatic, hepatic and post-hepatic jaundice. Now that's a topic of pathology. Then what jaundice does to your body, the discoloration of the skin, sclera, mucosa and interstitial fluid. That's a topic of physiology. Then as I teach all of these in a story fashion, I'll be using lots and lots of diagrams which are a part of human anatomy. And that's how I'll be integrating everything together. So all you have to do is relax, take a deep breath and listen to my lecture like a story. Visualize whatever I'm drawing like a movie. So that's all you have to do. Starting off with bilirubin. Your red blood cells, they have an average lifespan of 120 days. After 120 days, the young red blood cells have become old and they get destroyed. See, the young red blood cells, they are flexible. But the old red blood cell, they have lost that flexibility. They are no longer flexible. Just like humans, we youngsters are quite flexible. But as we grow older, let's say we reach the age of 50 years, our joints, they become rough, our muscles stiff, blood vessels hard, back rod-like stiff. So the whole body loses flexibility as we grow older. Similarly, red blood cell as well, when it is young, it is flexible, but after 120 days as it has become old, it loses that flexibility. It loses its flexibility. Okay. So 120 days have passed. The red blood cell has become old. It has lost its flexibility. Now it needs to get destroyed. Where does the red blood cells get destroyed? There's an organ in the body which is called the graveyard of RBC, because majority of the old RBCs get destroyed there. You know what that organ is? That's none other than your spleen. So this old red blood cell, it enters the spleen. It's not just that only old red blood cell enters the spleen. The old red blood cell, the young red blood cell, blood as a whole, they enter your spleen. Now inside spleen, you have the spleen capillary, which contains gaps. These gaps are referred to as sinusoids. Now hear me out. If this is a young RBC, it is very flexible. See, this is a young RBC. It is very flexible. So it can easily squeeze through this gap, come out, and then continue into the circulation. But this RBC right here is old. It has lost its flexibility. So it is unable to squeeze through the sinusoid. So it is stuck. It is stuck or trapped into the sinusoid. Now this stuck or trapped old RBC that no longer has the properties of young RBC gets phagocytosed by the macrophage of your spleen. This macrophage is a part of reticuloendothelial system macrophage. Reticuloendothelial system includes the macrophages of three organs: the macrophage of spleen, the macrophage of bone marrow, and the macrophage of liver. So the old RBCs can get destroyed by the macrophage of spleen, the macrophage of bone marrow, and the macrophage of liver. But since majority of the old RBCs get destroyed by the macrophage of spleen, that's why spleen is regarded as the graveyard of RBC. Okay, I hope till now you have understand. You had your young RBC. After 120 days, it becomes old and loses its flexibility. So inside the spleen, there are capillaries with sinusoids. The young RBCs can pass and squeeze out of these sinusoid and continue into circulation. But then the old RBC, it is quite stiff, so it is unable to squeeze through the sinusoid. It gets trapped in the sinusoid, and then these trapped old RBC that no longer contain the properties of young RBC gets eaten up by the macrophage of your spleen, which is a part of reticuloendothelial system. Okay, so this in blue, I have zoomed into the macrophage, and inside this macrophage is none other than our RBC. Now let's break this RBC into its various components. What is RBC? It is a bag filled with hemoglobin. The bag being none other than the plasma membrane. What is plasma membrane made up of? It is made up of lipid and protein. So when lipid gets broken down, we get free fatty acid, and when protein gets broken down, we get amino acid. Plasma membrane, done. Now what is inside RBC? Cytoplasm. Cytoplasm contains number 1, enzymes. Tell me one enzyme. G6PD, glucose 6-phosphate dehydrogenase. Have you heard of G6PD deficient patient or G6PD deficient anemia under hemolytic anemia? I've already taught you this under the topic of anemia, for which I have made a single video explaining all the concept and all the types of anemia. Was that out after this lecture? Okay, so there's enzyme. The enzyme gets broken down into amino acid as well. Finally, what remains? What remains is hemoglobin. Let's break down hemoglobin. You know the structure of hemoglobin, right? It contains 4 heme and 4 globin. 4 globin. Out of 4 globin molecules, there's 2 alpha globin and 2 beta globin. Be it alpha or beta, they are at the end of the day what? Protein. And protein is broken down into amino acid, and then the amino acid gets reused. Now we are just left with heme. Do you know the structure of heme? Heme is made up of 4 pyrrole molecules. What? 4 pyrrole molecules. 4 pyrrole molecules. 4 pyrrole molecules connected with one another with the help of methane bridge. Methane bridge. And in center, there is iron. Again. So what do we have? We have 1, 2, 3, 4 pyrrole molecules connected to one another with the help of methane bridge, forming a single ring, which is called the porphyrin ring, and a central iron. So let's degrade this as well. Let's break it down. So first, the iron from the center is snatched out. Okay, so we are taking out that iron. Iron snatched out. And this iron is then what? It is stored. It is stored in the form of what? It is stored in the form of ferritin. The ferritin polymerizes. The ferritin polymerizes to form a larger molecule, which is called hemosiderin. So the iron snatched from the heme of hemoglobin is stored in the form of ferritin, and ferritin polymerizes to form a much larger stored form of iron called hemosiderin. So both ferritin and hemosiderin are stored form of iron. Okay. Now what remains? What remains is the ring of the heme of hemoglobin. The ring of the heme of hemoglobin gets degraded, and what we get is biliverdin. And then this biliverdin gets reduced, and what we get is bilirubin. Now there are 2 enzymes required here. Number one, for the degradation of the porphyrin ring into biliverdin, and number two, for the reduction of biliverdin to bilirubin. So the first enzyme which degrades the ring of the heme is heme oxygenase, and the second enzyme which reduces biliverdin is biliverdin enzyme for reduction reductase. And finally we get bilirubin. The term bilirubin is there. So what is bilirubin? Bilirubin at the end of the day is the end product obtained from degradation of hemoglobin. So the hemoglobin present in the RBC gets degraded and broken down, finally giving us a waste product. So the end waste product obtained from the metabolism or degradation of hemoglobin is bilirubin. Okay, another concept. The bilirubin that we get from the degradation of hemoglobin is unconjugated bilirubin. Few properties of unconjugated bilirubin. Number one, it is unconjugated. Number two, it is lipid soluble. Number three, it is non-polar. And number four, it is toxic. Remember the 4 points. It is unconjugated, it is lipid soluble, it is non-polar, it is toxic. See, actually the property is just two. One, that it is lipid soluble, and another that it is toxic. It is lipid soluble because it is unconjugated, and lipid soluble are non-polar. See, these two properties are responsible for two things. Number one, since bilirubin, unconjugated bilirubin, is lipid soluble, it can easily enter inside tissue because lipid soluble substances can easily penetrate through the plasma membrane. So the unconjugated lipid soluble bilirubin can easily enter inside tissue. Second, since it is toxic, once it enters inside the tissue, it will destroy the tissue. So you don't want two things from happening, okay? You don't want two things from happening. See, bilirubin produced in the macrophage of the spleen from the degradation of hemoglobin is unconjugated. Since it is unconjugated, it is lipid soluble, that is, it loves lipid. And lipid soluble substances, they can easily cross through the plasma membrane of the cell. So since bilirubin is lipid soluble, it will cross through the plasma membrane of the tissue. But this bilirubin is what? It is a waste end product. Waste. It is toxic, so it will destroy the tissue as well. So we don't want two things. Number one, we don't want this bilirubin to enter inside the tissue. And number two, since it is toxic, what does our body do to toxic substances? It excretes it out. So our body needs to excrete out the toxic unconjugated bilirubin. How does it do so? See, this is how. So finally, from the degradation of hemoglobin, we get unconjugated bilirubin. This unconjugated bilirubin from the macrophage is thrown into body circulation. It is thrown into body circulation. Now this unconjugated bilirubin is in body circulation. Let's draw it. Unconjugated bilirubin. There is tissue in the periphery. Tissue in the periphery. What did I say? Unconjugated bilirubin is lipid soluble, so it can easily cross through plasma membrane. Unconjugated bilirubin, it is lipid soluble, it can cross through the plasma membrane and enter the tissue. It is also toxic, so it will destroy the tissue. But why does this not take place normally in our body? In our body, continuously old RBCs are getting destroyed, hemoglobin is being degraded, and bilirubin, unconjugated bilirubin, is being produced, which is being poured into body circulation. Unconjugated bilirubin is being poured into body circulation continuously. But then why is it that this unconjugated bilirubin not enter into our body tissue and destroy it? What is preventing that from happening? That is none other than the fact that this unconjugated bilirubin inside body circulation circulation binds with a plasma protein. It binds with a plasma protein, albumin. So what? So this. See, unconjugated bilirubin, it is lipid soluble, plus it is small in size as well. So it is easy, it can easily enter inside tissue. But see now, this unconjugated bilirubin, which is bound with plasma albumin, is now too large in size. And this large molecule can't penetrate through the tissue membrane. It can't pass through the tissue membrane. So it is now present within the circulation. It will not enter into any tissue of the body. First problem done, right? So first thing we didn't want was for bilirubin to enter inside tissue. So that's done. Second thing we want was for body to get rid of this toxic material. And there are two ways our body gets rid of any toxic material. I mean, there are many ways, but two major ways our body gets rid of toxic material. Number one, via feces or stool, and number two, via urine. So how does that happen? Let's see. Okay. Under general pharmacology, you might have already learned that the drugs that we take are mostly lipid soluble. These lipid soluble drugs, once it has been metabolized, once it has been used, it can't get excreted out. First, it needs to go through the liver, where it gets metabolized, so that the lipid soluble drug is converted to water soluble drug. And then this water soluble drug can then get excreted. This lipid soluble drug is the non-polar drug, and the water soluble drug is charged or polar. So the main concept is that first, the unconjugated bilirubin, which is lipid soluble, which is non-polar, needs to be converted to water soluble, which is polar, and only then after that can it get excreted. So it has to go through the process of conjugation. Here is the liver. Here is unconjugated bilirubin. First, the unconjugated bilirubin enters the liver. The first step, uptake. Then unconjugated bilirubin is converted to conjugated bilirubin. As a result, lipid soluble bilirubin becomes water soluble bilirubin, or non-polar bilirubin becomes polar bilirubin, or toxic bilirubin becomes non-toxic bilirubin. Non-toxic bilirubin, the process is called conjugation, and finally it is excreted, it is secreted into body, and then it can get excreted through feces and urine. Okay, so let's see these three steps. What's the diagram here? This whole thing that you see right here is representing a liver, but more specifically a liver cell. So this is your hepatocyte. This is the systemic circulation. This right here is the enterohepatic circulation, the portal system in between liver and the gastrointestinal tract. You know that from your anatomy, right? Between gastrointestinal tract and liver, the blood does not directly go to your heart. First, from the GIT, the blood goes to liver and then to your heart. So in between, that extra circulation is there none. That is portal circulation, or hepatic portal circulation, or the enterohepatic circulation. Entero referring to GIT and hepatic referring to liver. Okay. The liver makes bile. The bile, it goes through the bile canaliculi present within the liver, and then through the right and the left hepatic duct, then the common hepatic duct, which fuses with the cystic duct, forming the common bile duct, which fuses with the pancreatic duct, forming the hepatopancreatic duct, which opens into ampulla vata, which is guarded by the sphincter of Oddi, which opens into duodenum. Which part of the duodenum? The second part of duodenum. You must know your basic anatomy. Duodenum has four parts. The first part, the second part, the third part, and the fourth part. It's in the second part of the duodenum where the biliary system opens. Okay. With that basic anatomy done, let's see what happens to unconjugated bilirubin. So the unconjugated bilirubin gets carried by the albumin to your hepatocyte. This unconjugated bilirubin is then taken up by the hepatocyte. The process, the very first step, as I said earlier, is referred to as uptake. It takes place by facilitated diffusion. Now that unconjugated bilirubin is present within the hepatocyte, it goes through what? It goes through conjugation, and the unconjugated bilirubin becomes conjugated bilirubin. Second step, conjugation takes place. For conjugation to take place, something has to come and conjugate with bilirubin, unconjugated bilirubin. What is that something? There's an acid. The acid is called 2-UDP glucuronic acid. Glucuronic acid. So that gets conjugated. Obviously, an enzyme is required for the metabolism to take place, and the enzyme is none other than 2-UDP glucuronyl transferase, because the transfer of glucuronic acid is taking place. It's the glucuronic acid that is getting conjugated with the bilirubin. So glucuronyl transferase enzyme is required. With that, conjugation is also done. So finally, you have conjugated bilirubin. This conjugated bilirubin, it has two fate. Fate number one, it enters into the enterohepatic circulation. Conjugated bilirubin enters into the enterohepatic circulation with the help of multi-drug resistant protein 3. Okay, but then this conjugated bilirubin, which entered enterohepatic circulation, is taken back into the hepatocyte. It is taken back, reuptake takes place, and that is done by OAT1B1 and OAT1B3. So two pumps, multi-drug resistant protein 3 and OAT1B1 and 1B3. Remember the name of the pumps because you will require them under the topic of congenital hyperbilirubinemia. Okay, so that was the first fate. The second fate is secretion. The third step, it gets secreted into biliary system. And that takes place with the help of another pump, that is multi-drug resistant protein 2. This step is referred to as number 3, secretion. So secretion takes place. Finally, the conjugated bilirubin is inside the biliary system. From the biliary system, where does it go? So hepatic duct, then the cystic duct, again the hepatic duct, the cystic duct, and then inside the gallbladder. Inside the gallbladder, you have conjugated bilirubin. What does gallbladder include? It includes bile. What is bile made up of? It is made up of 3 major stuff. Number 1, bile salt and bile acid. Number 2, the substance that makes up the bile salt and bile acid, that is cholesterol. And number 3, whatever is here, conjugated bilirubin. So 3 stuff, bile salt and bile acid. Number 2, the stuff that makes bile salt and bile acid, that is cholesterol. And number 3, this right here, conjugated bilirubin. The conjugated bilirubin then repasses through the cystic duct, then the common bile duct, then the hepatopancreatic duct, then the ampulla of Vater, passes through the sphincter of Oddi, and then gets secreted into the second part of the duodenum. So finally, the conjugated bilirubin is present in the second part of the duodenum. The conjugated bilirubin inside the gastrointestinal tract, it is acted upon by what? It is acted upon by bacteria, the bacteria of the gastrointestinal tract, and gets converted to urobilinogen. The urobilinogen that is formed by the metabolism of conjugated bilirubin by the action of gut bacteria has 3 possible fates, and they are: number 1, see, the gastrointestinal tract has a connection with your liver via portal circulation. The blood from the heart goes to your gastrointestinal tract, and from the gastrointestinal tract, it does not return directly back to your heart. It goes through extra piece of circulation that is present in between, and that is the portal circulation or the enterohepatic circulation. Entero referring to the intestine, the GIT, hepatic referring to liver. So nutrition, bunch of things from the GIT gets absorbed into the portal vein, the portal circulation. Similarly, the urobilinogen produced also gets absorbed into the portal circulation or the enterohepatic circulation. Urobilinogen. From enterohepatic circulation, it reaches from the entero to hepatic, that is from GIT to liver. And once it reaches the liver, what does it do? It traces back its pathway. That is, it enters the biliary duct, the biliary canal, and return back to the duodenum where it initially was. But not all urobilinogen return back, cause there is one more connection with liver. That is, all the deoxygenated blood from the liver, it comes to the heart via portal vein. So some urobilinogen, it returns through the biliary duct, but a lot of it goes back through the hepatic vein to your heart, and then from heart the systemic circulation, and from the systemic circulation to your kidney. And it is in the kidney, let me show the connection directly from here. So in the kidney, urobilinogen is oxidized to urobilin, and urobilin provides urine its characteristic yellow color. Okay. What about the urobilinogen present within the GIT? It comes out as well. Yes, it comes out along with stool. So in stool, urobilinogen, it gets oxidized to stercobilin, and stercobilin provides stool its characteristic brown color. So that's it. That's the end of the story. Let me revise the whole thing for you with the help of a flow chart. So what do you have? You have initially the hemoglobin. The hemoglobin is made up of heme and globin. Globin is broken down into amino acid, which gets reused. All of this is happening in the macrophage of reticuloendothelial system, especially your spleen. The heme contains a central iron and a porphyrin ring. The central iron is stored. It is stored in the form of ferritin. Ferritin polymerizes to form hemosiderin. What remains is the porphyrin ring. The porphyrin ring gets degraded into biliverdin. The degradation of the porphyrin ring into biliverdin, or the heme into biliverdin, takes place with the help of enzyme hemoxygenase. Biliverdin is then reduced. It is then reduced to bilirubin. Biliverdin is reduced to bilirubin. This takes place with the help of enzyme biliverdin. Enzyme required for reduction, reductase. Now, this bilirubin that is obtained from the degradation of RBC, the degradation of hemoglobin in the macrophage of reticuloendothelial system, is unconjugated bilirubin. This unconjugated bilirubin comes into systemic circulation, where it is carried by albumin. So here is unconjugated bilirubin, which is carried by albumin. Plasma protein binding albumin, which takes it to liver, where the very first step of conjugation takes place, which is called uptake. So first step of conjugation, uptake, takes place. So it enters the liver. Inside the liver, the unconjugated bilirubin gets converted into conjugated bilirubin. So this is the second step, which is conjugation. Conjugation takes place, I get it, but conjugation with what takes place? Conjugation with 2-UDP glucuronic acid takes place. Since conjugation with glucuronic acid takes place, so the enzyme required for this metabolism conjugation is 2-UDP glucuronyl transferase, obviously. Now this conjugated bilirubin has two fate. One, it can enter into the enterohepatic circulation. What? The enterohepatic circulation. Via what? Via multi-drug resistant protein 3. But then it returns back as well. It returns back with the help of what? OAT 1B1, OAT 1B3. See here. So inside the liver, conjugation of the unconjugated bilirubin into conjugated bilirubin takes place. Then this conjugated bilirubin can enter back into the enterohepatic circulation with the help of multi-drug resistant protein 3, which is but taken back with the help of OAT1B1, OAT1B3. But then the major fate of conjugated bilirubin is that it enters the biliary circulation with the help of multi-drug resistant protein 2. It enters the biliary circulation, and this is the third step, that is secretion into biliary circulation. From the biliary circulation, the conjugated bilirubin enters the duodenum, where the conjugated bilirubin gets converted to what? Urobilinogen. Now urobilinogen has 3 fate. Number 1, go back into enterohepatic circulation. See, urobilinogen in the duodenum can go back into enterohepatic circulation, but then it returns back, stresses back the same thing, and then returns back. So it returns back. But not all of it returns back. Does all of it return back? No, some of it enters the systemic circulation and reaches the kidney. In the kidney, urobilinogen gets converted to urobilin, which provides urine its characteristic yellow color. And the urobilinogen present within the gut comes out along with the stool. In the stool, it gets oxidized to stercobilin, and this stercobilin provides stool its characteristic brown color. That's it.